Hello, everybody. I'm Valerio Piomponi. I'm really glad to be here today and have the chance to introduce myself to the PRP community and also shows which is my background, what are my expertise, and what are my perspective for the work to be done here in Area Science Park. So I'll start maybe giving some information about myself. So my background is in physics. I study at the University of La Sapienza, with also 1 year mobility to Grenoble, where I have my internship in an institution called LIFI that proposes research on the biophysics fields. And then I moved to Trieste, where I had my PhD at SISSA, where I work under the supervision of Giovanni Bussi in the Faculty of Physics and Chemistry of Biological Systems. And since the 1st of December, I started my new position here at the Area Science Park, in particular in the lab of data engineering. And of course, my position is funded by the PRP project. That's why I'm here today. So today I will mainly like to talk about my background. So I'll give a brief overview about my PhD project, and then in the end I will give some perspective about what I want to do in the future. So my PhD project that I just finished in the last October was about using molecular simulation to investigate the impact of post-transcriptional modification on RNA structural dynamics. So I will start giving a brief introduction on RNA, which is one of the most important molecules in biology and is mainly and historically known to be the molecule which transfers the genetic code from DNA up to protein through the process of transcription and translation. However, RNA can play many other fundamental roles in the cells through the so-called non-coding RNA, like for example, transfer RNA or ribosomal RNA. And to understand the function, it's really important to look at the structure, like for protein. But since RNA molecules are really flexible molecules, it's really important to not look at single structure, but rather at the structural dynamics of RNA. One of the most powerful computational tools we have to characterize three-dimensional structural dynamics of biomolecules, it's molecular dynamics. This is possible by making use of empirical force fields that are a list of parameters governing bonded and non-bonded interaction in this classical and relatively simple functional form, was still able to capture the main physics of biomolecules. So in this representation, basically the interaction between atoms are described by this classical potential. For example, a covalent bond is described by a spring. So you need to derive the parameters K that give you the stiffness of the spring, and so on. You can characterize angular springs or torsional spring, and non-bonded interaction are generally described by Lennard-Jones or classical electrostatic potential. So the way you derive these parameters are several. In general, they are fitted on quantum mechanics calculations. So quantum mechanics calculation, of course, are more accurate, but they are possible only for very small systems, small fragments. So what you do is basically you fit your classical model on quantum mechanical models for very small system, and then you can export the classical model also to bigger system and simulate for longer time scales by simply integrating the equation of motions given by this functional form. Of course, molecular dynamics, it's just a model, and it has some limitations. To give some examples, this functional form cannot describe for the polarizability of atoms. Also, this model cannot describe chemical reactions because you cannot destroy the springs, for instance. So you need, of course, quantum mechanics to describe chemical reactions. And more in general, we can say that the accuracy of molecular dynamics is limited by the quality of the force field. So how good you derive these parameters. So in principle, you should always validate your simulation on experiments, or maybe also corroborate or integrate your simulation with experiments. So there's two main methods to do that. One is to also fit the force field on experiments. in such a way that the force field are also transferable to other systems that do not belong to the training dataset. And the other way is to do ensemble refinement with methods like maximum entropy or maximum parsimony. And this method consists in modifying the probability distribution predicted by molecular dynamics as small as possible in order to enforce some experimental evidence that you know about. Another limitation of molecular dynamics is its computational costs that make it difficult to sample big systems for long time scale. And these slides try to resume which are the typical RNA timescales and also what can be tackled with standard computational methods. And with molecular dynamics is usually generally reasonable to get to the timescale of microseconds, maybe milliseconds, but this could be not enough to sample interesting stuff like conformational rearrangements of RNA structure. But this kind of limitation can be tackled using enhanced sampling techniques that are sophisticated methods that allows you to accelerate your system and enhance the accuracy of their events. This method can be classified into classes. There are methods that allows you to heat up the system, so you bring your system to higher temperatures. At higher temperatures, more interesting things can happen, and then you go back to low temperatures and do your statistics about the system. Then other methods consist in adding a bias along a reaction coordinates. This method can be much more efficient, but a proper reaction coordinate must be known, and this is not always the case. It's not trivial. So the topic of my research was RNA modifications. So let's talk about this. Normally, people know about RNA as a polymer molecule, which is composed by 4 types of units that are the standard nucleotides: adenine, uracil, guanosine, and cytosine. So normally, a 4-letter alphabet is used to describe RNA sequences. But in reality, things are much more complicated than this, since a lot of modification exists. They can be artificial, of course, but also natural modifications that happen in the cell after transcription. There's enzymes that can write this modification, enzymes that can recognize this modification, or the enzymes that can erase the modification. And the modification consists in simple biochemical modification of the nucleobase. Like, for example, here we had a methylated adenosine in this position. That's called M6A. And N1A is another adenosine that is methylated in this other position, and so on. These are just few examples, but there's more than 100 modifications that are known to exist in nature. And these modifications are widespread in the cells, and they have important functions. They can affect RNA folding, and they can affect the target specificity of RNA interaction. So the purpose of my project was to try to use molecular simulation to investigate the impact of this modification on RNA structural dynamics. But of course, a problem we need to face was the problem of the force field. Indeed, along the years, a lot of effort has been done to parameterize at best the 4 standard nucleotides, but not a lot of effort has been done so far for modified nucleotides that are so many. So we cannot really trust the force field for modified nucleotides, also because they are not being validated against experiments yet. So we must need to take care of this and always try to combine our simulation with experimental data that are available for modified nucleotides. Okay, these slides give an overview about the project that I have overseen during my PhD, but today I would like just to talk about a specific one that is a project in which we use the ensemble refinement techniques to combine our simulation with experimental data for a system containing inosine, that is a common modified nucleotides. So the adenosine to inosine editing that consists in like removing an amino group from the adenosine, replacing it with an oxygen. This process is widely studied. It's known to that its regulation affects immune response, and also it's known that the inosine to uracil base pairing introduce flexibility on double-strand RNA. And for that reason, our collaborators that are experimentalists from the University of Munich decide to investigate a specific system that is a 20 base pair RNA helix, where 4 inosines are present in the central part of the helix, each of them base pairing with uracil. So they perform several measurements on this system. In particular, they perform NMR measurements, and they had interesting data concerning this NMR J couplings data that were indicated increased C2 endo population in the central part of the helix. The C2 endo is a possible conformation of the sugar puckering, so this part here of the nucleotides, that is alternative to the C3 endo conformation. And the C3 endo is the one expected in double-strand RNA with the standard canonical A-form helix. And these J-coupling signals can be mapped against torsional angles, a specific torsional angle that is related to these two hydrogens, H2 prime and H1 prime. So you have a specific forward model called Kaplan's equation that allows you to relate the NMR signals to these torsional angles. So you can use this model to also back calculate the signals from a molecular dynamics trajectory, for example. And from this plot, you can see that high values of the J-coupling signals correspond to C2 endo conformation, whereas low values of these signals correspond to C3 endo conformation. Another interesting measurement they perform is small angle scattering experiments. The data were indicated an increased radius of gyration of the double-strand RNA with respect to the standard one with no modifications. So this data was interesting, so we decided to use molecular dynamics simulation to try to reconstruct an ensemble of structures which is compatible with these experiments and look in with deeper detail how these structures look like. To do that, we use several sophisticated methods. We also use an enhanced sampling techniques called replica exchange collective variable tempering. I won't go into details here because it's really technical. I'll just say that we need this because we were interested in this sugar puckering conformation. So we wanted to ensure an exhaustive sampling with respect to this conformation. And we use this technique to enhance the switching between the C2 and the C3 endo conformation for all the nucleotides in the helix. Then once our simulations are done and we have our trajectories, before analyzing them, you can also try to refine, do the refinement by using this method, this principle called maximum entropy principle. The goal here is to improve the accuracy of our prediction by incorporating some experimental data in our prediction. So this method consists in finding a probability distribution that is as close as possible to the prior, where the prior is the one predicted by molecular dynamics, but among those which are compatible with experimental averages. Here in the box, I'm giving the formulas for those who are familiar, maybe. It just consists on maximizing the relative entropy by putting some constraint on the observable you want to match with experiments. And then you find a probability distribution that is related to the prior. And the problem is just finding these lambda parameters by that can be found by an optimization procedure by maximizing the entropy. We apply this principle to 9 J-coupling signals that are related to 9 different nucleotides in the structure. And also we apply this on the square radius of gyration that is predicted by SAXS experiment. So in order to enforce the right radius of gyration. This plot gives you an example of what happens when you apply maximum entropy to a specific J-coupling signals. So the green line shows you the probability distribution of these signals as predicted by molecular dynamics. You have a very big peak for low values of the signals, and a very small peak for high values of the signals. And you obtain this average given by this vertical dotted line, but the experimental data correspond to this blue dotted line. So you want to enforce this average. And what you need to do is like to rebalance the two peaks in order to get the right average. So these are the methods Now I'll try to show some results of our investigation by comparing 4 different samples that we generate. So the gray one stands for the adenosine ensembles, so correspond to the system without inosines, to have a reference of a standard double-strand RNA. The red ensemble I correspond to the inosine ensembles as predicted by molecular dynamics. The blue ensemble is the one in which we also enforce NMR data. In the green case, we enforce both NMR and the radius of gyration predicted by SAXS experiments. The first plots show the population of canonical pairings in the double strand. So as you can see, the adenosine to inosine editing introduce the, flexibility and dynamic pairing in the central part of the helix. This evidence further increased when you also enforce experimental data. The second plot shows you radius of gyration distribution also. This black bar corresponds to the experimental value. As you can see, the red ensemble, the one of inosine, highly underestimated the experimental value. But if you enforce the NMR data, you're going in the right direction of improving this radius of gyration. And of course, you can enforce it by performing maximum entropy also on the SAXS data. Another interesting thing to look at was the population of this C2 endo conformation for the nucleotide in the central part of the helix. And as you can see, in the red case, the population are really low. You cannot even appreciate them. But when you enforce NMR data, this population increased by also 1 or 2 order of magnitude. And that means that the molecular dynamics and the current force field we are using highly underestimate the C2 endo population. And we really need to enforce them with the maximum entropy. We can also look and investigate to what extent the adenosine to inosine editing introduced flexibility on the double-strand RNA. I'm showing that by showing these two-dimensional density plots with respect to the twist and kink angle. So the kink angle tell you how much an helix can bend, whereas the twist angle can tell you how much a helix can untwist, for example. And we can appreciate here that the inosine editing allows you more untwisting of the double-strand RNA. And then when you enforce NMR data, so you induce this C2 endo population of the sugars, you also allow for more bending of this double-strand RNA. Then these colored figures I'm showing here, I'm calling them flower bouquets. I produce them by aligning 100 structure extracted from the ensembles. And this is to qualitatively show how much this ensemble looks rigid or flexible. So the gray one seems really rigid and regular, whereas the inosine ensembles look much more chaotic. And also I can quantify the ensemble RMSD with respect to these alignments. And this value dropped by 1 order of magnitude in the inosine case. Another thing that I tried to do was to look for conformers in these ensembles. And I did that by performing principal component analysis. And this analysis allows us to individuate 5 basins, 5 clusters here. And I'm showing this cluster with this dynamic secondary structure representation where the color of this arrow show you how much the bindings are populated. So the darker they are, the more they are populated. So if you look at these conformers, B1 and B2 looks like standard A-form helix with canonical pairings, whereas the other 3 conformers, B3, B4, and B5, show like dynamical pairings and non-canonical pairings also. For example, if you look here, you have these black arrows. That means that this contact is 100% populated, and it's a contact between 2 inosines that are not aligned in the sequence, that are in a complementary position. I will go back to this later. First, I want to say a few more words about another important aspect of this investigation, that is the validation of our ensembles. So we use some experimental data to refine the ensembles and to generate ensembles. We can use different experimental data to validate them. And we use noise data that once again comes from NMR measurements, but these times are data that are related to distances between protons, whereas the J-complex that we use were related to torsional angles. So we can check how much our trajectories agrees with these noise distances, and we can see that our refinement ensemble have better agreement. So lower chi-square means better agreement. We can also look at which kind of signals get in agreement only when we do the Mach-Cent refinement. These are the signals that are listed in this table. They are mostly related to distances that are intranucleotide or intrastrand, but there is also an interstrand distances related to inosine 30 and inosine 12. And this is really interesting because this contact is the one we could observe from molecular dynamics that is shown here. It's a really strange contact, and it seems like that the noise data are validating this strange conformation that we could observe in the simulation. Another validation we can do is on the SAXS spectra. So I remind that we use in ensemble refinement only the radius of gyration as information coming from the SAXS spectra. This information extracted just by looking at what happened for low values of Q in this spectra. So what happened here, the curvature here. Then you can fit on the whole spectra, and we could observe that the fitting works better in our refinement ensembles. So just to resume this work, in this work that I decided to present here, we show how molecular dynamics, in particular the current force field that we were using for these inosines, works bad, highly underestimate the C2N population in the system, meaning that the parameters for inosine should probably be fixed. But we also show how a combination of molecular dynamics with enhanced sampling techniques, and also in particular by integrating data coming from NMR and SAXS experiment, can be used to construct ensembles that are in much more agreement with experiments. And then we also shed light to what extent this adenosine to inosine editing induce significant dynamics on double-strand RNA producing conformers with non-canonical pairings and on common helical parameters. This is all for this project. I just want to show a last slide that kind of resume what I'm interested in or what I would like to do in the future, what are the ideas. So I still want to stay in this field of investigating biomolecules, structural dynamics, combining different methods. Biomolecules mean that not only RNA, could be protein, could be DNA. It also can be like RNA protein complex, molecules that are binded to ligands and so on. So I've shown how molecular dynamics can be used, which are the advantages of molecular dynamics. For example, the fact that you can really have unlimited resolution. You can look at all the atomic details, but also shown which are the limitations. So the problem of precision, that is related to the problem of sampling of possible conformation of specific molecules, but they also show how this limitation can be tackled using enhanced sampling techniques. And the other limitation of molecular dynamics is the accuracy due to the quality of the force field or bad quality of the force field sometimes. But that also shown how this limitation can be solved by integrating the simulation with data. Data that can come from experiments. And here I'm listed the experiments that are generally used, that are generally combined with molecular dynamics. So X-ray, you're not really combining with molecular dynamics because X-ray usually give you a single structure, and this structure is usually used as a starting structure for the simulations. Then you can guide your simulation related with information coming from NMR or SAXS experiments that are solution experiments. Okay, so give you information that are time average, that correspond to molecules that are in solution and not in a crystal, so they're much more complementary, let's say, to molecular dynamic simulations. And also in the last year, cryo-EM information are widely used, also in combination with molecular dynamics. There's a lot of people that it's working on these techniques, on new techniques to combine data that come from cryo-EM that are generally electron density maps. And you can check if these electron density maps are reproduced by molecular dynamics, for example. Then there's also the field of artificial intelligence. So in principle, you could also use data coming from the artificial intelligence to combine with molecular simulations. The artificial intelligence method can be of several types, but what they generally do is they can predict information about molecules, like for example, the native structures, just starting from the sequence of a protein, for example. Maybe you all know about AlphaFold 2, which is outperforming any other method to predict the native structure of proteins. But for RNA, there's not yet an AlphaFold 2. There's still a lot of people that is trying to work on this problem for RNA, but it's much more difficult to have something really reliable for RNA structures, for predicting RNA structures. There's several reasons for which this is so difficult, but one of the reasons is, as I said, is that RNA is really flexible. Often the structural dynamics is important rather than a single structure. So one of the questions that people is asking is, can this artificial intelligence method can be used also to predict information about not only a single structure, but alternative structure, intermediate structure, and more in general about the dynamics of molecules. So we are also trying to propose this question to ourselves. In my lab, there's a lot of people that is pushing toward the direction of using artificial intelligence, in particular language models, to study biomolecules, and we want also to start to go in the direction of using these methods to predict information about dynamics of molecules. And in that case, we could combine with molecular simulation in first instance to validate the prediction of the models. And then if the answer is yes, they can predict information about dynamics, we could also think about integrating this information in molecular dynamic simulations to integrate them. So I'm already start to work in that direction, in that area of combining simulation and artificial intelligence. And then I'm looking forward also to find new chances also to collaborate with experimentalists to tackle some interesting challenge that biology can offer. So thank you for your attention.